We're live. Good morning, everyone, and welcome to Act 3 of PISCeS 2026. For those of you who are able to make the reception last night, it was good to meet new faces and reconnect with old friends. For those of you who are just getting here this morning, buckle up. We've got a pretty exciting program. My name is Bob Hanner. I'm the director for the Biodiversity Institute for Conservation Synthesis here at the University of Guelph. It's my pleasure to welcome you. And without further ado, we'll get started with our first keynote talk from Charlyn Partridge, the Annis Water Resources Institute at Grand Valley State University, on using eDNA to investigate hemlock woolly adelgid in eastern hemlock ecosystems. Welcome, Charlyn.
It should have been, so sorry that it's not.
No worries.
Oh my god.
It's large. There are lots of pictures, sorry.
I like a presentation with a lot of pictures, so.
They're very pretty. There are forests. I do.
At least the connection is fast.
Yes. I can draw y'all a tree.
All right.
Beautiful. Awesome. Perfect. Thank you so much. Good morning. Thank you for the invitation to talk to you today about the work that we've been doing over the past few years, trying to use airborne eDNA to monitor for a very tiny invasive forest pest in Michigan forests. So everyone can hear me, right? We're good on that front. Okay, perfect. So imagine you, your friends, your family, you're taking a walk in the forest. You're on the trail and you look over to your right and you see something like this. So where you see a little archway and then behind that archway is just darkness. Now imagine you're brave enough to walk off that trail and then walk into this dark patch in the forest. Does anyone know what you're going to be surrounded by when you walk in that patch? Anyone have a guess? Hemlocks. What you will find is you will find yourself surrounded by eastern hemlocks. So the environment within an eastern hemlock forest is very different than the environment outside of an eastern hemlock forest. And this is because hemlocks are what are known as foundation species, where they have a disproportionate regulation on the environment in which they're found in. And so they regulate things like light. They have a very thick upper canopy that prevents light from penetrating the understory. Because of the fact that they block out so much light, they regulate temperature. If you're in the woods and you walk in a hemlock stand, you can literally feel the temperature decrease. And then when they drop their needles, this actually impacts the soil chemistry, causing a drop in pH in the soil. And then also because of their evapotranspiration rates, they regulate how much water that ecosystem can hold. So when you lose hemlocks, you actually drive ecosystem level changes in these environments. And because of this strong regulation, the ecosystem in a hemlock stand is very unique. So they have very unique biological communities. In Michigan, we love our hemlocks because this makes up a large proportion of our forested area, especially in the Upper Peninsula. In total, our state has over 170 million hemlocks. Although these forests are being threatened by a tiny invasive species known as hemlock woolly adelgid, for the purpose of this talk, I'm going to refer to it as HWA. So it's a tiny invasive forest pest that infests eastern forests in both the US and in Canada. And this is an image of an adult. So they have this long proboscis, this long mouthpiece that they actually feed into the twigs of the hemlock. It goes in and then the individuals feed on nutrient cells. So they suck the nutrients out of the tree. This results in a decrease in photosynthesis, and then these trees can die with anywhere between 4 to 10 years. And so the image that I'm showing you now is one of the trees in one of our study sites that has died. So here's the trunk of the tree, and then the upper part has broken off. And then this image to your, whichever, right, is basically me standing at the base of that tree, holding up my camera, showing the upper canopy. And what you can see is after that tree fell, you now have a large gap in that canopy where light can now penetrate. So this is one tree, but now imagine a forest where we've seen the devastating impacts of HWA result in up to 95% mortality in eastern hemlocks. And so and this is an image of an eastern hemlock forest, and all of these brown trees are dead hemlocks due to HWA. And so when you lose this strong regulator of this environment, what happens is you have complete ecosystem shifts. So what was a coniferous forest is now a deciduous forest because of these major changes in this strong environmental regulation. So HWA was introduced to Eastern North America around prior to the 1950s from southern Japan. The original point of infestation was around Richmond, Virginia, and since that introduction, it has spread through the majority of the eastern hemlock distribution range. So we have populations all the way down in Alabama, up to Nova Scotia, and then populations from Virginia all the way to Michigan. So within that 7 years time span, it has rapidly expanded. And the only areas where we don't find it is at the most northern extreme of the hemlock distribution range. So a lot of our work is in Michigan, because that's where I'm housed. And so we'll talk a little bit about HWA in Michigan. Our current infestation was first discovered in 2015 in Allegheny County. That's this most southern area here on this map. All the red dots are known HWA infestations. And then our current known infestation reaches all the way up to Antrim County. And so basically all of the counties along the western edge of Michigan are now infested. And this is not great because the response, Michigan's response to HWA when it was first detected was very aggressive. So they started doing surveys across multiple areas near where infestations were found, sending out people to actually look for it. Whenever they found a tree, they immediately treated it and then created a buffer zone. So treated trees within an 800-foot buffer zone to try to slow down the spread. Yet despite that, we've seen significant spread over the past 10 years or 11 years. And we're very worried because now our known infestation is right near the Upper Peninsula. So it just needs to jump across the Mackinac Strait to get to the Upper Peninsula, and that's where our highest density of high quality hemlocks are. So when we talk about HWA monitoring, one of the things that when we started getting on this project, we wanted to make monitoring more efficient and then faster. And so from talking to our partners, specifically, this all started through a partnership with the National Park Service. They came to us and they were like, hey, we heard of this method called airborne eDNA. Do you think this would work for hemlock woolly adelgid? And I was like, no idea, never done it. Let's try it. And so I was already working with them on a different invasive species. I do molecular stuff, so I was like, And so we tried it. Up until then, the main way HWA is monitored is you take a group of people, you bring them out to the forest, you're in a stand, and you say, look for HWA. So they systematically look at trees, look at branches, and see if they find HWA. And what they're specifically looking for are these little white dots on the needles of hemlocks. So this is called an ovisac. In that ovisac is an individual. That's where they lay their eggs. So a couple of problems with this, with visual surveys. Number one, HWA infestations typically start at the top of the canopy. Hemlocks are very tall and work their way down. So you're not going to see those initial infestations if they start at the top. Second thing is these ovisacs are more robust during winter. And so you're sending field crews out in hemlock forest in Michigan in the middle of winter trying to find white tiny spots on trees. Anybody think of a problem with that? So Michigan, kind of snowy, also very cold. And then also, Michigan has 170 million hemlocks. Like, if you do the math for how much time you actually spend surveying each tree, that's like 120 years to survey the 170 million hemlocks we have. So to kind of make this process more efficient, we wanted to see if we could use airborne eDNA. So I'm not going to define airborne eDNA here because we all know what it is. But through our lab, an undergrad student in my lab developed these 3D printed traps. So this is what we use. These traps have a base and a lid, and they hold 4 microscope slides that have been dipped in petroleum jelly. We put them out in the field, so this is an image of what our trap would look like. And then we collect those slides every 2 weeks and test for the presence of HWA. And so right now, our monitoring program is centered around high-risk sites. So these are sites that our partners have identified as being high risk. And we monitor them from May to June, so those traps go out through the entire, what we call the crawler period, so the most mobile stage of HWA. We exchange those slides every 2 weeks, and then we use qPCR to target a 250 base pair region on the cytochrome oxidase gene, and then we look for presence absence of HWA. And so this is just kind of, all the stars indicate where we have series of traps out right now. So right now is our monitoring season. So this is kind of where our traps are distributed throughout the state. Here's kind of our workflow. So we bring the slides back to the lab, we scrape off the petroleum jelly into a tube. We do a series of heating and centrifugation steps to separate out the material from the petroleum jelly. Then we take that material, extract the DNA out of it, and then test it using qPCR. So each sample is run in triplicate, and then for us, all of our samples, all of those triplicates have to come back positive for us to actually call something a detect. So, and the reason, and I know this is more stringent than a lot of cases, but the reason for that is because when we say that there is a detection, that triggers a cascade of events. We call our partners, we say, here's the GPS location. They go out and spend significant amount of time and personnel surveying that area to find those infested trees. So if we say it's there, that's thousands of dollars out of their pocket that they need to go spend and survey. So when we say it's there, we want to make sure that it is there. And so this is just kind of like one of what our output looks like. So we have a target threshold based upon our negatives. Oops. One. And then here is what 2 positive traps would look like. So we have all 3 of our replicates coming out positive, and then we have our HWA control. So how has this worked? We've been doing this since 2022. So the inaugural year, when we first tried it, we had 55 traps out. 2 of those came back positive, indicating 1 new stand where HWA was. In 2023, we identified 3 new stands. In 2024, we identified 1 new stand. And then we got a larger grant in 2025, built up more partners. So we have like 200 and something traps out right now. And last year, we identified 8 new stands of infestation. And so what this map is showing, hopefully you can see it, is kind of where those traps are relative to where the infested trees that our partners then went out and found. So for 2022, the 2 traps that came back positive are these yellow triangles, and then the trees that were infested were these pink dots. And then that circle around that pink dot indicates that 800-foot buffer where all trees will now be treated. And then for 2023, the red triangles are our traps that came back positive, and then the green circles are the trees that came back to be infested. So overall, this program has been positive. So we now have detected the first known infestation of HWA in a national forest in Michigan. Last year, we detected the most northern infestation that we know of in Michigan. So this is something that we feel is beneficial, but more than us, our partners think this is beneficial. So this is just a news story indicating the first known infestation in a national park. This was here on Manistee National Park. And we weren't interviewed. These were our partners who gave us the shout out that this was found because of eDNA work, right? I just received an email from another partner that was like, you gave us a positive. We went out and surveyed 1,500 trees in that acreage, and we found the 2 that were infested. So this method is really, really sensitive, and it really works, or it works really, really well. And so far, anytime we've said we get a detect here, they have gone out and they have found it. So we think it is one thing I want to point out is though it's not a replacement for surveys. Like we're not saying this should replace surveys. This works in tandem with surveys. HWA is a needle in a very, very large haystack, right? So when I say tiny, HWAs is the size of a grain of sand, right? So what we do is we say, here's your haystack, a forest. Search in this corner of that forest because it's probably there, right? So that's kind of how we see this working in tandem with their visual surveys. So it narrows the search range that they have to do. So that's our current monitoring program. Another thing that we want to do with these traps is we wanted to see, can we use eDNA or the data from our traps to not only monitor high-risk areas, but give some information about infestation levels. Because this could be important for our management groups if you have a high infestation versus a low infestation. That might change how they prioritize sites for treatment. And then also, while these treatments, the chemical treatments that they apply are effective, they don't last forever, right? So a lot of the treatments tend to wear off after 5 years. So if you can monitor a site continuously across years and start to see when HWA is coming back up again, that might signal a time when a site needs to be retreated. So that's kind of how we see using quantitative assessment may come into play for specifically HWA. And so to do this, all the work that I'm talking about from now on is work that's been done by my former grad student, Keely Dunham. But to kind of see whether or not we could use this as an indication for infestation levels, we looked at 9 sites with known infestation within them. These sites ranged in infestation levels. We had some that were low infested, some that were really high, and then they ranged in their treatment dates as well. And so the way we did this is we wanted to compare using our trap or qPCR methods, so these newer methods, to standard methods of estimating HWA density, which is using something called random branch sampling. Okay? So random branch sampling basically counts the amount of HWA cystins on a subset of trees in a forest. So before we start, I'm going to give you a crash course in HWA life history. So HWA has 2 generations per year within its infestation range. It's clonal, so all of this is done asexually, and those life stages consist of eggs, crawlers, and cystins. So an egg or crawler will hatch from an egg. This is the most mobile stage when it's most likely to move to a different place, and then it will settle, go through a couple of instars, and become a cystin. Then that cystin will live on that tree for the rest of its life. So, and then that cystin will lay eggs, that will hatch into crawlers, that will then move and then settle into cystins as well. So the cystins are the individuals that are living on that tree for the rest of their life, okay? So the way cystins counts works is you go to a stand and you take a subset of trees in that stand, and then you take a subset of branches on that tree, and then you count the number of cystins on a portion of that branch for each of the branches, for each of the tree, for each of the sites, right? So in our case, we had 9 sites, so we went to those sites, we randomly sampled 10 trees, then we randomly sampled 9 branches, and then we counted the number of cystins on 100 new growth needles on each of those branches. Mathematically, that's over 90,000 needles. So this is not something that's fast. This takes months to actually assess infestation levels. But if we can do this using trap from our data, and that trap is pretty indicative of infestation levels, then this really speeds up that process, right? And so basically our cystins counts were what I just explained, and we did that from fall through winter of 2022. And then from our traps at these heavily infested sites, not only are we just capturing eDNA that's present in the environment from HWA, in the heavily infested sites we can actually catch individuals. So we can catch individual crawlers, their most mobile stage. And so that's what, oh, sorry. Giving away all my answers. So in addition to doing cystin counts on the trees, when we exchanged the slides out, we would count the number of crawlers on each of those traps. And then crawlers should theoretically be related to cystins because your crawlers are the offspring of the cystins, right? The crawlers laid the eggs, or sorry, the cystins laid the eggs that developed into crawlers, so you should have a good relationship between those two. And then, so for each of our sites, we had 5 traps deployed, and then we assessed those crawlers over that period. And then for qPCR, we then scraped all the petroleum jelly off those slides and tested that for the presence and quantified the amount of DNA that we had. So the first thing I'm going to show you is the qPCR to crawler count data, because this should be highly correlative, right? The DNA is literally coming from the individuals on the slides. So we should have a really strong relationship. And so this is looking across all of those sampling periods. We had 7 sampling periods across all 9 sites. On our x-axis is the crawler count, on our y-axis is the gene copies, and we had a relatively strong relationship. So if you do ecology and you get an R-squared 0.7, you're like, I am awesome and my lab is beautiful, right? And so we got a pretty good relationship when we looked across all of the sites together. So next, we wanted to pull sites apart and kind of look at them individually, and then we were sad because that relationship doesn't really hold up when we look at individual sites. And specifically, when we are looking at our low-infested sites, we typically have a lower correlation in our low-infested sites like Golden Township and Spalding compared to our more highly infested sites like Barbara here. So we do have a strong overall relationship, but there are some issues. So this can't be a predictive measure, right? So you can't, because it's very site dependent, how strong that relationship is. Part of this could be variation in DNA extraction efficiency. Part of it, especially those high infested sites, we're getting a lot of extra airborne eDNA from ovisacs and hemlock needles and stuff like that. So all of that is contributing to some of the noise that we're seeing. So the next thing we wanted to do is we wanted to see whether or not, if we just looked at this data during the peak crawler period. So remember that data before was across all 7 of our collection periods, so the whole time that crawlers are out. But crawlers peak at a very defined period. Normally the first week of June is when the majority of the crawlers are out. So we wanted to see if we just look at that peak crawler period, do we get a better relationship, because that's when most of those crawlers are present. And so that's what this is looking at. So we're looking at crawler count versus cystin count from that random branch sampling, and then cystin count to qPCR values. And what we see here is a pretty good relationship, so summed across all of our sites. So each point is one of our sites, looking at that single peak crawler period at each site. And so again, we're getting that really high correlation, looking at crawler counts to cystins and qPCR to cystins. So what does this mean in total in terms of management? Can I use qPCR or crawler counts to tell you exactly how many cystins you have in a forest? No, I cannot tell you you have 284,637 cystins in your forest. Can't do that. Can I tell you that you have a really high infestation or a really low infestation? Maybe. And then a lot of times that's all the managers want to know. Do I need to tackle this now? Or can I wait? Or do I tackle the low infested area because it has the highest chance of eradication? Right? Those are the kind of decisions management needs. Or it can I tell you you have a trending increase or a trending decrease? Possibly. And this is important, especially for managers who have limited funds but need to treat sites, right? They don't want to treat unless they have to. So if we're seeing an increase, then that could re-trigger when it needs to be retreated. So is it perfect? No. Can it be used to estimate exact number of individuals? No. Can it be used in management under certain situations depending on the information that you need? Yeah. It might be a cheap, fast alternative to give you a general idea of if you have a lot of it or a little bit of it, right? So the last thing I'm going to talk about is some of the work that we are doing looking at whether or not HWA can help, or eDNA can help us understand ecological shifts that are occurring in these hemlock forests. So, like I said before, when you start seeing hemlock decline, your environment changes drastically. And so this is an image of just a standard hemlock forest. This isn't these are not the same areas. I don't want to give you that assumption. But this is an image taken from David Orwig, who's been looking at HWA for decades and looking at environmental shifts in the in Harvard forests due to HWA. This used to be a hemlock stand, right? So this used to look exactly like a hemlock stand. But now what you see, because of that opening in the opening in the canopy, allowing light to reach the understory, you're getting lots of shrubs and brushes, you see an increase in plant biodiversity, you see an increase in arthropod biodiversity in these settings with hemlock loss. And so we had these traps already out in these areas for monitoring. So we had traps monitoring as to whether or not we could look at infestation levels in our infested sites. We had traps distributed based upon where our partners wanted them in high-risk sites. So we had sites with HWA and sites without HWA. Already our traps deployed. And we knew that we caught DNA from other things because you could look at it and you could be like, there's tons of pollen on this trap. Or my students would be looking at it and there's like an insect leg. So we knew we were getting DNA from other things. So we were like, well, can we sequence this and figure out if there are different communities present in infested versus non-infested areas? And so that's what we did. So this was kind of like a proof of concept thing. So here are our infested sites. Like I said before, we had 5 traps per site. Here are our high-risk sites. The numbers vary per site because this is based upon what our partners want. Our partners send us shape files in GIS. We put 1-acre grids over those areas, and then we distribute traps based upon how many they want to create good spatial resolution. So that's when the number of traps varies in those high infested sites because that's solely dependent upon where our partners want to monitor. Right? But in total, we had 100 traps deployed throughout the season, and then we had 7 sampling periods throughout that time. And so our workflow is very, very similar. Extract the DNA from the slides, except this time we did amplicon sequencing where we targeted arthropods and plants communities. So for the sake of time, I'm only going to talk about the arthropods. Also, we found no difference in plant communities, so that data gets a little bit boring. So I'm going to focus on our arthropod data. And so I should say we had 7 collecting periods, but for the amplicon sequencing, we pulled all those collecting periods together. So we had 1 sample per trap that we ended up sequencing. But that sample represented collections from May until end of July. So this is a bubble plot showing kind of the most abundant, or the top 20 most abundant genera that we found on our traps. So in total, we identified 321 unique genera that encompassed 190 families. So the way this bubble plot works is here are our high-risk sites, here are our infested sites, Here are the site names, the size, and then here are the genera that we're talking about. So the size of the bubble represents its relative abundance in that sample, and then the color is reflective of the family of that genus. So if you see two that are the same color, then that means that these two groups belong to the same family. So maybe unsurprisingly, our most abundant arthropod was Adelgis, which is Adelgits. We happen to see more Adelgits in infested sites than in high-risk sites, yay us. And so that's kind of what you would expect. But the thing I really want to point out is we were getting Adelgits, which are tiny things. We were getting moths, which are bigger things. We were getting midges and mites, which are tiny things. We were getting bark beetles, which are larger things. It's not on here, but we were getting daddy longlegs. So we were getting a DNA from a range of sizes of insects. So when you look at that slide looks clean. Like it looks like Vaseline with a little bit of black dots on them. But when you actually sequence it, we're actually getting a lot of DNA from a lot of different arthropod species present across various size ranges. And so, like I said, our prediction was that because of the loss of HWA, because of the loss of hemlocks, we would predict to see higher biodiversity in arthropods because of the shift in those vegetative communities. It's what we predicted. So here at the top, this graph is showing both alpha and beta diversity. So at the top, well, not technically alpha, we're looking at observed genera on our top one. So here we have our high-risk sites and our infested sites. Again, our site names are on the X. And so what it's showing is that statistically speaking, there's no significant difference in alpha, in the number of observed genera between our infested and our high-risk sites. But I want to point you to these three sites here. So this is Minart Park, Fruitland Park, and Pioneer Park. These sites are our three sites that are not treated. So the other infested sites are infested, but they're treated. So we're just seeing a trend in higher number of genera in our untreated sites than either our treated sites or our high-risk sites. So this could be a couple different things. It could be a treatment effect. The treatment does impact nicotine receptors, which is known to be a general pesticide. But when they treat trees, they actually drill holes in the hemlocks and inject the hemlock itself. So these aren't like basil bark sprays. You're literally treating the tree. So theoretically, anything that's not feeding directly on that hemlock shouldn't be impacted. Also, one thing I want to note is that the treatments actually help the hemlocks. So even though these sites are infested, they're relatively healthy because they've been treated. These sites that haven't been treated, this was in 2023, they were dying. I went to those sites last week and they are dead. Like, there are almost no hemlocks, live hemlocks left in those sites. So we think maybe what might be driving this is a combination of treatment effects, but also the hemlocks in this area just aren't healthy, right? So they're losing canopy cover. You are maybe starting to see some of those ecosystem shifts. So we didn't see it across the board, but we are starting to see some of the predictions that we had when you start to lose hemlock. And we think that is at least part of what's driving this. And then this bottom graph is looking at beta diversity. So this is looking at differences in arthropod communities. So this is a principal coordinate analysis. If two dots are close to one another, then that means that their arthropod communities are similar. If they're apart from one another, that means that their arthropod communities are different. The circles represent our infested sites, and the triangles represent our high-risk sites. And the ellipticals represent 95% confidence intervals. So what this is showing is that there's a distinction. So there's non-complete overlap between our infested sites and our high-risk sites, suggesting that there are differences in communities between those infested and high-risk sites. So when we look at what is different between those sites, we did a differential abundance analysis, and we identified 40 different genera that were different between these sites. I'm going to focus on 3 because Those are the most interesting in my brain, and y'all have to listen to me. So the three that I'm going to talk about, the first one is probably obvious, right? We saw more HWA in high infested sites. Okay. The other one is, or the other two are a little bit more interesting. We saw high numbers of spongy moth and Asian lady beetle in infested sites compared to non-infested sites. These are both also invasive species, right? So it could be because of the ecosystem level changes, but when you look at the literature, one of the things that to me is really interesting is spongy moth larvae preferentially feed on HWA-infested hemlock than non-HWA-infested hemlock, and larvae that feed on infested hemlock grow faster than those that don't feed on hemlock. So what that means is the presence of HWA may actually be making the population of spongy moth worse, and there may be a preference of spongy moth to infested HWA. So it's not the change in hemlock, it's actually the presence of HWA that's driving some of these differences. And same with lady beetle, or Asian lady beetles, they are predators on HWA-X. So it may be the presence of HWA driving these changes as opposed to the hemlock loss. And so, in summary, we feel like using airborne eDNA is an effective monitoring method for HWA and other tiny invasive forest pests that might be hard to see. One of the things that we think is a benefit is that it allows for continuous monitoring over the entire summer and kind of saves money from sending out survey crews to random places that you don't even know if it's there. And then also coupled with metabartic coding approaches, you can possibly gain even more information about these environments as well. And so with that, I just want to thank all of my partners the partners are amazing, and this would not be able to be done without them. So for all of our partners through all these years, huge props and thank you for all the work that you do. They're the ones treating trees, carrying big backpacks up like 90-degree ravines to treat HWA and every single hemlock in those areas. So they're doing the majority of the work. But then also to my lab and all the work that they've done as well. So thanks.
Do you think the fact that the spongy moth is having preferential sort of infestations where there's HWA is just because the HWA are weakening the immune system of the trees?
Yeah, so I think that's exactly what it is. So there have been studies that have shown that when trees are infested with HWA, because of the fact that they don't have a lot of energy anymore, they can't make a lot of chemical defense compounds, so it makes them even more susceptible to other things coming in. So, yeah.
That's kind of terrifying.
Yeah, I know, right? It's like a double whammy.
That was awesome.
Thank you.
What did it do without the microphone? Thank you. Yeah, this is really relevant for this audience as well.
It's better with a microphone.
It is better?
Yes.
Okay, sorry. Hey, for Ontario at least, most of our old growth forests are hemlock.
Mm-hmm. Yeah,
So they're easily the oldest trees, at least from Sault Ste. Marie south.
yeah.
And you're probably aware of certain increased fire risks.
Mm-hmm. Yeah.
Apologies for the air quality. So this is very, very relevant
Yeah.
and interesting. Have you looked at sort of what's the spatial sensitivity
Mm-hmm.
of your traps?
Yeah, so we do have the spatial sensitivity of the traps. It's one of the first things that we did, and it's about an acre, so that's it's about the size of a American football field, right? So, yes, and I was like, I know the but anyway, so that's why when when managers give us the properties that they want to survey, we put a 1 acre grid on top of it, and then put a trap per grid, or a lot of times we can't do a trap per grid because they're looking at 300 acres, right? So we kind of put as many as we can. Yeah.
Sorry, one more question.
Yeah.
is in the Great Lakes, there's a parallel, they call it an invasional meltdown. Tony Ricci already coined the term. Are you seeing any signs of other invasive species beyond the big three you mentioned?
So,
Or this could be facilitating other invaders?
yeah. So in terms of arthropods, not necessarily, but I would not be surprised if as soon as that understory gets light, any of the invasive plants that are in those areas are just going to take off as well. Hemlocks are great at suppressing biodiversity. So, you know, something that comes in and is a great like initial introduction colonized takeoff plant is going to explode in those areas. So, yeah, unfortunately. And there have been other studies that have shown similar things, kind of like in the more northeastern part of North.
I think we've got a cap questions there. Keep on schedule. Thank you, Charlyn.
Thank you.
We have Dr. Robert Moise from University College Dublin talking about integrating molecular tools into freshwater ecological status assessment.
Yeah, you'll just use the arrows. All right, so good morning, everyone. I'm Robert from UCD in Ireland, and today I'm presenting part of my PhD research on developing a new tool for freshwater ecological status assessment. So, as a general overview, hydrogen is the name of the project. It's an EPA-funded project in Ireland, and we are using both traditional and genetic methods for informing the integration of these new methods into water quality and biodiversity monitoring. So we have sampled 40 different sites in our country for 2 years. 36 were extensive sites that we monitored once every year, and the 4 intensive sites, we sampled them once every season. And we look at invertebrates, microbes, fish, and diatoms. So in Ireland, we have the Irish Q-value index, which is based on macroinvertebrates, which are divided in 5 different groups, A to E, according to their sensitivity to pollution. And the proportion of these invertebrates indicate the water quality. So normally the Q-value ranges from 1 to 5. It has been used for many years, well known in our country, but it's expert-based and difficult to standardize. On the other side, we have the DNA methods, which are non-invasive, low-cost, highly sensitive, and they're really flexible. And we're currently employing DNA metabarcoding, looking at the CO1 region and using 2 different databases, RDP and BLAST. So in terms of my samples, normally we collected water for eDNA analysis. Then I did the kick sampling, so I collected invertebrates that I used for morphological identification. And I stored that sample in pure ethanol for at least 1 month. After that, I filtered the ethanol and got DNA from that source as well. So here I have different photos. Again, collected water, 2 liters, 3 replicates per each site. Collected invertebrates for the Q value and the ethanol source at the end. So in terms of results, here I have intensive sites year 1. Here I show the distinction between water and ethanol source. You can see how these methods are quite complementary. And in terms of the taxa detected, the water eDNA recovers a broader signal, while the ethanol is linked to the actual kick sample. And apparently, seasonality has an underestimated role. So as you can see, depending on the time of the year, you could get more species using one method or the other. Here I show how the different groups, A, B, C, D, E, are distributed between the 2 different methods. And normally, eDNA has a lower overall richness, but recorded a stable signal. Normally, the group C is the more abundant one, as it includes tolerant freshwater species that are distributed across the country. We switch now to something interesting, I think. So the ethanol source, I think, represents the ideal approach, as you can get the best of both worlds. You can get molecular data, and it also has a defined sampling location compared to water eDNA analysis. And I think that could be really interesting for the future, as you can get consistency across sites, operators, and it's also really nice because you can get more enhanced taxonomic resolution, and it's useful for long-term data monitoring or retrospective analysis. So here's the formula that I've been working on lately. Is calibrated on the intensive sites, and it looks at the different groups in terms of taxa detected, and each group has its own weight. Now, in this formula, I'm not looking, and I didn't use the read abundance, just the number of taxa detected, and you can see there's higher values were generally associated with better ecological status. You can see how the Q value classes were distinguished, and the different weights for each class. Now, this formula has been tested on the extensive sites, and again, you can see there's agreement with the traditional ecological assessment status. Right now, it's more a continuous metric. There are no formal thresholds or classes, but that's something I would like to do in the future. And lastly, as I said, I've been not looking at the number of reads, only at the taxa detected, but I think that's still interesting information there. So I was thinking of developing these maps that are based on the read abundance profile as a complementary tool, where you can have, so then you can have your index and maps like this, where you can see the distribution of the reads across the different classes. So thank you very much for your attention. I have a poster number 3. I hope to see you there. Maybe we can chat about it. Thank you.
Next we have Laura Dutheil from the University of Guelph, who will be speaking to us about barcoding versus morphology, assessing different methods for invertebrate identification and determination of industrial effluent effects.
Okay, hello everybody. I'm going to be presenting the research that I did while I was doing my master's here. Thank you for the introduction. So in my title here I have the word EEM, so you're probably wondering what that is. This is environmental effects monitoring, and you can kind of think of this as an environmental assessment that is done on any active mines and paper mills in Canada. So this is a government-regulated thing that has to be done to make sure that the impacts are mitigated. What this looks like is a survey of the impacted area, the waterways, in this instance, for example, a river, or the exposure site, and comparing it to what we're seeing in an unimpacted site nearby, or the reference site. There are many components to EEMs. The part that I'm going to focus on is the benthic invertebrate component, and this includes morphological identification of the inverts done at the family level, and then assessed and compared between the reference and exposure sites along for these 4 endpoints, which are density, taxonomic richness, Bray-Curtis, and Simpson's evenness. So the goals for our research were to first see, can we do these EEMs assessment using DNA barcoding and compare that to what we would normally do with our assessments using morphology. So using both methods at the family level and running our analyses on the EEMs, can we get the same results? Can we use both of these methods? The other piece is that now that we have this molecular data, we are able to get a higher degree of taxonomic resolution. So we may be able to get down to the species level, or in the case that I'm going to be talking with you guys today, the bin level, which you can think of as a species proxy if you're not familiar. And will this change our EM results, right? So we're hoping that at the family level barcoding, family level morphology, that we see the same thing, but will we be seeing something different if we go down to the bin level? So very quickly, our methods were we sampled 5 sites in Canada, mines and paper mills. And we identified our inverts both morphologically and molecularly using DNA barcoding. So our results. There's a lot to look at here, but what I want you to pay attention to is each row here is one of our sites, and our columns that I want you to focus on are these 2 over here. So our first is our family level identification done using morphology. The other one is done using barcoding. And what we wanted to see is when we determined if there was a significant difference between reference and exposure sites, would we see the same conclusions using both of these methods? So for this example here, we're seeing that both of these methods are determining that there is a significant difference between the reference and exposure. So we have consensus here. Here as well. Here we have consensus again, but in a different way. So we have both methods determining that there was no significant difference between sites. Here as well. Here there is an exception, there was no consensus, and then consensus again. So what that leaves us with is 7 out of 8 of the sites that we studied, we see consensus between these 2 methods, which is fantastic. That ratio value you can think of as a 0.875, which we see represented in the first bar of this graph here. So that was our density endpoint that I just walked you through, but now we have those 4 other, the whole 4 endpoints. So you can see for our richness and Bray-Curtis endpoint specifically, we have 100% consensus. So all 8 out of 8 sites, we got the same results using barcoding and morphology at the family level. Another way to look at it, and again, this is kind of a lot, but you can think of, we're looking at 2 endpoints here. So the top one is Bray-Curtis, the bottom is Simpson's index, and what I want you to pay attention to first is these 2 sets of bars. These are our 2 family level assessments, so barcoding and morphology. So again, we're seeing that there's a lot of match between these 2 things. We can also now look at our teal colored boxes, which are our bin level values. And you can see that while we have that consensus between the 2 family level assessments, our bin levels are showing quite a bit of sensitivity and change from those. So our conclusions are that DNA barcoding and morphology can both be used and comparable in EEMs at the family level, and there's continued work on this in EEMs. Please check out Yomel's presentation tomorrow at 2:30, which will talk more about this. We also know that bin level analyses do provide more sensitivity, and our sister research also talked about how not all species within a family respond the same way to effluent. Thank you so much. If you have any questions, please feel free to send me an email, and thank you to all of the wonderful people who have helped with this research.
Next up, we have Colin Pattison from the Southern Alberta Institute of Technology talking to us about blowing in the wind, detecting terrestrial mammals using air samplers and nanopore technology in Alberta.
Thank you. We met last year at Cubes. Yes. Okay, good morning, everyone. I'm Colin. Over the past year, we started a small lab in Calgary to work with undergrad students in eDNA. And I'd like to talk about this project that we started originally to detect Franklin's ground squirrels, which I understand are particularly secretive, so difficult to find, and also declining in Alberta. So you're wondering why there's an elk on the picture there, and that's because after we started, of course, we decided to expand and look for more mammals. So we thought, why not try to capture airborne DNA and test our methods around red squirrel middens, which are pretty active all year long, and we thought would provide a steady supply of eDNA in the air around their middens. So at the time, my research assistant, who was Olivia, also wanted to test capturing airborne DNA from spider webs. And it was something that we had seen in the literature, but not in natural areas in Alberta, so I thought, why not give it a try? So we built this pilot project and set up these airplanes with the help of some undergraduate students. First in areas where we had lots of evidence of mammals, and then later on we moved these samplers to squirrel middens, and then we opportunistically gathered spider webs within a few meters of the air monitors. And in the wintertime, we switched a bit. So instead of gathering spider webs, we gathered snow where there were animal tracks, with the idea being that if there was airborne DNA, then it would land on the snow. So wherever we set up the airplanes, we also set up remotely triggered cameras. So then in the lab, we extracted with QIAGEN kits. We amplified with mammal-specific primers. We measured DNA quantity and quality, sequenced with Oxford Nanopore, and then we confirmed our classifications against the large NCBI NT eukaryote database. So I know that this is a pretty busy slide. The DNA concentrations were really low, so only about 0.1 nanogram per microliter. Overall, we produced about 35,000 reads that were classified to mammals. And I'm showing this to highlight that the snow and spiderweb samples gave us more detections than any of the other filters that we'd set up. So we also found interesting in blue that all of the filters detected wild boars, wolves, and muskrats, but we didn't detect any of those on the cameras. And it's also interesting that I'd previously detected wolves in these areas with cameras, but in more than 15 years of having these cameras set up in these areas, we've never detected wild boars or muskrats. The second part of that is that although we detected elk and red squirrels on the cameras, we never detected any of those in our eDNA samples. So to summarize, we detected carnivores, rodents, and ungulates, but overall our eDNA results were very low, and we did not detect any of the species, whether passive monitors that we did with spider webs and snow. The results from eDNA were kind of inconsistent with what we detected with the cameras, so that was interesting. So I think that this means that we've made a start in this area of detection, but we still have quite a bit more work to do to do more experimentation, to increase the number of species detections that we get, and to improve our repeatability. So I'd like to thank Olivia and our hardworking students, and thank you.
Next, and apologies if I get this wrong, Marjolijn, Rika, Nijzingh?
Kind of.
Will you please correct me?
Marjolijn, Rika, Nijzingh.
Thank you. From the University of Iceland, talking about implementation of environmental DNA into marine conservation practice.
Thank you. Good morning, everyone. I'm Marjolijn. I'm a PhD student at the University of Iceland, the Marine and Freshwater Research Institute, and MATIS. And my project is about eDNA, marine eDNA, and biodiversity research. So I'm looking at 4 different parts in my project. The first one is how marine biodiversity patterns around Iceland are structured across the different ocean basins. How do community compositions differ around Iceland, and if distinct bioregions could be identified. How, can citizen science eDNA samples be compared to samples taken by scientists? And how can eDNA data be implemented into systematic conservation planning? So we have 2 sets of samples. The first ones were taken during a survey by the Marine and Freshwater Research Institute in 2018, 2019, and 2025. That's the sample. The sample stations are on the left map. And as you can see, we have a pretty good coverage of the whole EEZ around Iceland. Samples were taken at 5 different depths, at 500, 250, 20, and surface level. Then there were citizen science samples taken during the Great Icelandic Swim of Ross Edgley, where he swam around Iceland with a support crew on a boat, and they took samples with a citizen science sampler, and these samples map you can see on the right side. So we use 3 different sets of primers. The first one is CO1. We use mitochondrial DNA for all different primers. We use the Leray fragment, and this one we chose specifically for the detection of invertebrates. Then we use MyFish, which is 12S. This is actually a mix of 3 different primer pairs, where we use U and U2 for fish, and E for elasmobranchs. And then we use Margar3, which is 16S, for the detection of marine mammals. My project is part of BioProtect, which is a bigger European project where we are designing tools to reach the 30 by 30 goal, and one of them is the Marine Planner, which is used for systematic conservation planning. This planner will use overlaying systems of data. So you could input fishing boat tracks, habitat. We want to use eDNA all the way to hydrography data. So we want to input alpha and beta diversity and species richness. So this is what the planner will look like. In the black and white, you see the grid that we will use, which is made out of hexagons, and based on the data coverage, we use very small ones or bigger hexagon sizes. So for input into the Marine Planner, you you need to input the conservation features, which can be habitats or eDNA data. Then you need to input cost, which is the quantitative value and relative costs of implementing conservation. You need to have constraints, which can be areas that are already in MPA or where there is economic activity which cannot be stopped. And these grid cells of the hexagons can then be locked for appointing a possible conservation area. And then you need to set your targets, which is based on percentages of how much percentage of this conservation feature you want to protect. This then looks like on the big map, you can see an example of a bay in Ireland, which is where they are making this tool, and all the areas that are green would be appointed as a conservation area, and this way you can really look at how much you want to conserve and what in what area you would need to look at to want to do this. Thank you very much for listening.
Next up, we have Christine MacPherson from the University of Guelph, who will speak to us about how anthropogenic stressors shape microbiome assembly in bumblebees.
Fantastic, thank you. Yeah, as mentioned, my name is Christine MacPherson, and I'm a PhD candidate in the Alan Berko Lab here at the University of Guelph. And today it's my pleasure to discuss with you how anthropogenic stressors affect bumblebee microbiome assembly. So to start off, as many of you are aware, bees are highly valuable pollinators and they pollinate most plants on Earth. They're responsible for 1/3 of the human food supply. Unfortunately though, many of these bees are in decline, and you might have seen headlines like this, and there's dozens more where those came from. And so, as for why these species are in decline, it's generally thought that this is due to habitat loss, infectious disease, as well as exposure to toxins and agrochemicals. But something important to note here is that it's not just the bees themselves that suffer from these exposures, it's the microorganisms that live within the bees. And so these microorganisms together constitute the bee's gut microbiome, and this microbiome is critically important for pathogen resistance, digestion, and the detoxification of environmental pollutants. And so because of eusociality, which has facilitated close contact, overlapping generations, and the sharing of food for millions of years, actually there's been a community of microbes that has become host adapted to bees and cannot be disturbed without having significant impact on the host. And for a healthy microbiome in bees, this is a balance of symbiotic microbes that resist colonization by pathogens and helps them to tolerate environmental stressors. But on the other hand, we can have a situation where the microbiome is degraded and is missing these important microbes, leading bees to be vulnerable to disease and toxins. And so to better understand how the bumblebee microbiome is affected by anthropogenic stressors, we performed a meta-analysis with the central question being, how does the bumblebee gut microbiota differ between natural and anthropogenic environments? To start this work, we collected bumblebees from 3 environments in Ontario, so that being laboratory, urban, and natural environments, as well as collecting bumblebee samples, we didn't collect them ourselves, but collected them online, for bacterial DNA sequencing data from all across the world, leading to us having about 814 samples, which is the largest microbiome analysis of pollinators to date. As expected, we found that habitat is the largest influencer of microbiota composition, so the level of anthropogenic stress that these bees encounter. And you can see here, this is an RDA plot showing, for those of you who aren't familiar, each dot is a microbiome sample from an individual bee. And you can see that natural environments, shown there in green, cluster differently from bees from agricultural and urban environments, as well as bees that are indoor controls or that are deliberately exposed to pollutants. We also found that wild bees hosted 253% more genera compared to their captive counterparts. And so this heat map shows on the bottom we've got different studies, including the work that we did here, and then on the y-axis we've got different microbial taxa. And with wild bees on the left and captive bees on the right, you can pretty clearly see that there's a lot of microbes that are not present in bees in captive environments. So we separated these, of course, and we found that there were some microbes that are present across all of the bees in our study, and so those are the microbes that we considered to be the core. We also found that there were some microbes present at about 50% prevalence or higher in wild bees, but they fell below that threshold in captive bees, leading us to call these ones captivity-sensitive core. Many of these are actually characterized as bee symbiotes. And then we also found that there were microbes that were entirely missing from bees in captive environments, and so these might have important roles for bee health that have yet to be explored. not only do bees exposed to the greatest amount of anthropogenic stressors have a different number of genera within their microbiome, the structure of that microbiome and the way it interacts is also different. So this is a network analysis. It looks a little complicated, but really the point is that each of the circles there is a different bacterial genre, and then the amount of green connections you have shows that that is a more cooperative microbiome. And so it's clear to see that on the left, the bees in the outdoor or the wild environments, those have a greater number of positive connections than the bees indoor, indicating that there is a more cooperative microbiome present. Also, something interesting to note here is that there are different hubs, so the taxa that have the most connections are different in bees from different environments. And so the key takeaways from this research are that human-altered environments significantly impact the bumblebee gut microbiota. Wild bees supported a substantially more diverse, richer, and interconnected microbiota compared to their captive counterparts. And this type of data can provide a baseline for interpreting signals detectable via eDNA, so that when we survey these environments, we can understand how pollinator health is being affected by anthropogenic stressors. If you'd like to learn more about this research, we recently published it in Science of the Total Environment. Thank you to everybody who made this work possible, and thank you all for listening.
Next up, we have Dr. Sharon Wang from the University of Guelph talking to us about using population size structure to predict temporal eDNA dynamics in an experimental mesocosm.
Hello, good morning everyone. I think folks can hear me, I'm pretty loud in general. So today I'll be talking about population ecology set of eDNA, and more specifically how we use individual-based whole eDNA deposition rates in combination with population size structure data to predict eDNA temporal dynamics in the mesocosm experiment. So previously in PISCeS 2023, I talked about my very first experimental chapter, which I'm sure a lot of you remember because it was a really great 5 minutes. But just as a recap, what we did for my very first chapter is we're really interested on the individual level how eDNA deposition rate changes throughout individual lifespan. So we grow Daphnia in individual vials, as you can see in the photos, across 4 different experimental conditions. The take-home message for this talk is that we found a really good correlation that is on the log-log linear scale to explain eDNA deposition using body mass alone as a predictor. So this in particular tells us when we're thinking about eDNA as sort of aggregate all the individual eDNA deposition combined together for population level estimates, we need to consider population variation, population body size variation, especially for species that has a very wide range of body sizes. In this case, we see there's a magnitude of difference throughout individual lifespan as an effect of body mass. So how do we use this parameter to actually generate population level prediction? Well, this is my second experimental chapter where we actually grow these Daphnia in much larger mesocosm scales with my very great undergraduate students. We collected a bunch of experimental factors, including biotic factors like temperature, DO, total nitrogen, and phosphorus. And we also collected algal density, and more importantly for this experiment, we collected data on Daphnia density as well as biomass using a photo adaptation of very good software. What you can see in this little photo here, each of the Daphnia as circled in the petri dish actually captured as a moving particle, and then the output actually gave us a measurement of the body mass. So this is really great for population size distribution. We also collected eDNA on site as sort of observation, which is used to compare to our prediction value. And we use a 40 micron as a pre-filter to ensure we don't capture any tissues or eggs or anything. And then we use a 5 micron filter paper aiming to capture really large, recently released DNA fragments. So what does our data look like? Well, so this is a lot on this photo, but I want you to focus on the brown circle that you can see with axes on the y-axis on the right. So this tells us the population biomass for each of the 6 experimental tanks that we have as a function of time on the x-axis shown as sample size. So overall, we see that in the very beginning, across all our experimental tanks, the population biomass is pretty low, and they grow really quickly and sort of plateaus near the end of the experiment. You also see there's a violin plot hidden underneath each of them. So if I blow it up a little bit, each of the violin plot actually shows the exact size distribution for within that sample. So we see in the very beginning, there is very low biomass, and those individuals actually grow larger. So in later samples, you see the range of the size distribution actually expands to larger body sizes, and those larger individuals reproduce, so we see this kind of shift, cohort effect on those population size structure over time. So this is great, because those data actually, in combination with the coefficient that we get from the very first experimental chapter, allow us to actually measure what is a predictive value from each of the individual within our sample. We can add those together, that is our predictive value. So what does that look like? Well, on the axis, again, we have time as sample size. Y is the total eDNA quantity number. We have the observed value in the solid triangle in brown, and then the predicted value is shown as in red circle. So the first thing I want you to see, there are two distinct blue triangle that's in here. This is where we actually had a positive eDNA detection, but we didn't actually capture any Daphnia using our traditional sampling method, particularly in the beginning when the Daphnia density is fairly low. And when we're actually looking at what is the predictability of our model compared to observed value, we found that a broken line analysis is the best, which means in this case, there is a certain threshold that's being identified in our model that is around, I think 4,700 copies, whereas on the log scale it's about 8.47. Below this threshold, we did not see any significant correlation. But above this threshold, we see a very good fit between our predicted value and our observed value with a slope of 0.95, which is pretty much as close to 1 as it can get. So this really tells us when we're thinking about using eDNA to predict population abundance, we might want to think about that at lower density, which is most naturally occurring in most environments, that eDNA may not be a very good fit. But once you're above the threshold, there is a predictability that is often can be found, but depending on your sample size and your experimental system. Thank you very much, and I'll be around for any questions.
Our last talk for the lightning session is Shauna Dworatzek from University of Guelph, who will be talking about community-based monitoring of vectors and disease in the Arctic.
Hi everyone, my name is Shauna, and today I'll be talking about a little bit of the work I did for my master's project. So the frontier of climate change and related ecological impacts is expected to occur in the Arctic, and this is because the Arctic is warming at an unprecedented rate, and this unprecedented warming is causing many species to move poleward. Inuit community members, specifically in Kudluktuk and Cambridge Bay, are already observing these changes in the Arctic, such as landscape changes related to warming, as well as changes more specifically related to our project, higher abundances and different looking mosquitoes than seen in the past. And the reason that these different looking mosquitoes and other biting insects such as black flies can pose a risk is because they're able to transmit a variety of diseases, and the ones we were concerned about are vectored nematodes, which infect caribou. And more specifically, we looked at Xueqi, which is transmitted by mosquitoes, and Onchocercus cervipedis, which is transmitted by black flies. And although neither of these directly cause death in caribou, both of them lead to decreased body condition and can have corresponding or cascading effects on herd health. So we worked with members of the communities in both these regions, Cambridge Bay and Kudluktuk, to develop a project. We worked with the hunter-trapper organizations in both communities, and this work started from the very beginning on planning the project, deciding what questions we were going to answer, as well as all the way to the end and collecting samples and sharing data. So in Kudluktuk, all the samples were collected by the hunter-trapper organization, and in Cambridge Bay, they were collected with guidance with the hunter-trapper organization, as well as you can see in this picture, there's me along with a community member, Hannah, and we collected the samples together in Cambridge Bay. So we did this across 2 summers in both regions using 2 different methods. And then we shipped the insects back to Guelph, where we sorted them before using 2 different primer sets. We used one general primer set to identify the insects, and another one which was very specific to the phalaroid nematodes. And then we used 3rd generation sequencing, or on them in ion. And then the reason that we did this is because we were thinking ahead into the future, and we wanted to try and use a method which could later be used in the Arctic. So we're hoping that this can later be adapted so that samples no longer have to be shipped down south, and they can do the entire research project in the Arctic. So we did not detect any of the parasites we were looking for, Xueqi or Onchocercus cervipedis, and we tested over 11,000 individuals. We also conducted a positive control analysis to ensure that our primers worked even with high concentrations of insect DNA and low concentrations of nematode DNA. So we're fairly confident that they're not in the insects in these regions at this time. And this is not entirely surprising or unexpected because the parasites thus far haven't been detected in caribou in Cambridge Bay, which is the further north community, as you can see on this map. And although they've been reported in Kudluktuk by community members and detected in the blue nose east herd, which is the herd found in Kudluktuk, as many of you I'm sure know, caribou are a migratory species. So this blue nose east herd also travels very much further south, and this even happens in summer regions, so it's possible that they're being exposed to these parasites further south near Great Bear Lake. So although we didn't detect any parasites during this survey, this isn't necessarily only good news. We did detect a high number of vectors in the region which are capable of transmitting these parasites, and with warming, these parasites are likely to become more prevalent in the future. And this is why continued monitoring and working with the Inuit communities in the region is so important to make sure that we can stay on top of the spread of disease to such a culturally important species like caribou in the future. Then I just want to thank everyone who made this research possible.
Let's have another big round of applause for all of our lightning talk speakers. Next, we have a workshop particularly of relevance to our early career researchers about making the most of your research, publishing, skill building, and career tips. This is presented by Genome, the International Journal for Genetics and Genomics, which is also a sponsor of this conference.
Thank you.
All right, can everyone hear me?
Please turn down the sensitivity, the reverb is very bad.
I'll let you do that.
Yeah. Okay. I don't know that I can adjust it. Yeah. Do you wanna, for this, maybe we'll get you on a handheld mic? I'll give that a try. No? Not yet? You have to hold it. It was. And it's up. There we go. Go with that.
Thank you. How's this? Better? Okay. So my name's Calvin. I am one of the managing editors for Biochemistry and Cell Biology and Genome. So these are our two not-for-profit molecular biology journals that I work on. And I'm also an assistant lecturer at the University of Alberta. I've kind of moved away from molecular biology research and more into research into science communication and publishing. Happy to talk about that at another time. A little bit about Genome. So Genome is a largely volunteer-run, not-for-profit journal. And why I wanted to talk about publishing with y'all today is I think everyone's already gotten a sense that publishing has become more expensive, right? Over time, there's more journals, but the cost of publishing is going up, and with how expensive molecular biology kind of already is to do, finding the right place to publish is an ongoing challenge for a lot of us. So I'm trying to pitch Genome as a potential place for you to consider. It's a not-for-profit journal. Open access fees are waived for all Canadian researchers. Everyone attending the conference can publish in the journal. Open access, completely free of charge. The point of researchers and academics like myself coming together to run a journal like this is to create a platform for other researchers to publish in where cost is hopefully not a barrier, right? We want to make publishing as accessible to everyone as we possibly can, and so again, the journal is completely volunteer run. Any profits that the journal does make, either through various workshops, opportunities, things like that, government funding. We redirect all of that back into supporting local conferences like PISCeS. So why does publishing choices matter more than ever, right? The number of journals has gone up. The cost of publishing has gone up. Whether or not journals are doing a good job at marketing and creating visibility for your research is a growing issue. There's also issues of ethics and transparency in publishing, especially in the molecular biology space. There's been growing issues of fraudulent research, fake data, AI-generated papers, and so finding a good journal for your home, for your research is important in preserving your own sort of integrity. So it's no secret that large-scale publishers like Elsevier, Springer Nature siphon a lot of money out of the research community, right? Their profit margins pretty much outcompete a lot of large tech companies. You can imagine publishing is naturally not a very expensive process, and so the fact that a group of volunteers can come together to run a journal like this is a good show of that. So if you're thinking of trying to redirect money into journals that support local conferences, Genome again is a good place to consider. But not just Genome, there's a lot of amazing society-run journals, not-for-profit publishers that run similar journals across various research topics. So again, something for you to think about when you're publishing, because your choices really do sort of impact the way publishing is done. It impacts it has an economic impact on the way funding decisions are made at the national level, and in turn, how other researchers in your community might be thinking about their publishing choices. So I wanted to talk a little bit about AI, because AI use in publishing has gone up. I know a lot of researchers are relying on AI to kind of help with the growing workload, myself included. So a survey done last year actually found that a large number, so over half of researchers are relying on AI to save time in one way or another. So either by doing sort of background research, manuscript preparation, developing course content, what have you. AI can often be a very good tool, but just a quick warning, a quick AI reality check. AI does come with flaws, right? It's not always providing you with the most accurate information. It can create unreliable sources if you're relying on it to help you with citation formatting, generating and finding reliable sources, things like that. It's also not always up to date in terms of information that it can provide for you, so that's something to sort of keep an eye out for. And of course, most AI programs that you might be using, like ChatGPT, is not really trained with scientific data, right? So again, it's prone to generating hallucinations of fake research data, fake citations that you might be relying on when it comes to doing your own research, and you find out later that it's not actually real. So quick tips when it comes to publishing: do not ever list ChatGPT or other AI tools as an author. Obviously, they don't have the human capacity to consent to having their name in a published article, and again, it's not a real human being. It cannot provide you with, it cannot verify the data, the data that you've generated. So do not rely on ChatGPT for that. Always apply human oversight, right? Again, AI is prone to generating fake content. It can be prone to generating fake sources. You do not want to be in a position where a journal has caught that you've used AI in your work without proper disclosure, because you can, it's tough, right? So I've had a case where and I know the authors well. I know the authors, I know the lab, I know they do amazing work. They're not the type of people who are going to be, trying to publish fraudulent work or fake content. But a student in the lab used an AI tool to help with formatting their citations. Some of the citations ended up being not real citations, and so when the journal and the peer reviews caught on to that, it was flagged as AI generated. We had no choice but to reject the work. I do know the group who submitted the work, and so I tried to advocate for them, indicating that I really don't believe, there was any malicious intent. They were using AI to save time in formatting references, but due to a conflict of interest, I'm not the one making the final decision, and the work was rejected for that reason. So you don't want to be using AI without proper disclosure. It just puts you in a tough spot. And I know no one in this room is going to be, trying to do anything unethical, but you just don't want to be put in that position to begin with. But if you are using AI, and that's not to say you can't use AI, it's best for you to just be open about it. So if you use AI to help with English writing, formatting, anything, it's useful to just indicate in the manuscript that you used AI for a specific purpose, so that if any errors are caught during the peer review process, we know that it's likely generated, by accident, and it was never, intended to mislead anyone. So I wanted to talk a little bit about sort of post-publishing activities. So journals actually do a lot of post-publishing marketing for your research. Again, not all journals are built the same, but I can share with you that Genome does put a lot of effort into making sure your work is visible beyond having it published. So, and I think this is particularly useful for grad students who may be kind of nearing the end of their degree, is to always ask the journal for additional visibility when you do publish with them. So request that the journal, do a social media shout out on your research, right? Include any personal lab handles so that they can tag you in it. Journals typically run X account, Blue Sky accounts, LinkedIn accounts. That's part of the journal marketing activities. So ask them to do more to make sure your research gets out there. Also has to be featured on the journal blog or newsletter, right? Journals run podcasts, journals run blogs, news sites as part of sharing research that's published in the journal. So if you ask, typically journals are more than happy to do that for you, especially if you're a graduate student, you're getting ready to defend, maybe you're looking for that next step in your career. Something like this could potentially catch the eye of potential future employers, future collaborators. I'll show you an example in the next page. And it also kind of shows how your research has impact beyond just the research community, right? There's people who may be working in policy, working with NGOs, government, who may see a blog post and find your work interesting and reach out to you for potential collaboration. So here's an example. This was research published last year, looking at sort of pheromones and how pheromones impact predation across a number of bug species in Northern America. And so after the research was published, they reached out saying, hey, one of my students is defending, this was their thesis work, would you mind doing a little bit more promotion around that? And of course, we were happy to do that for the student. Another example, not necessarily in Genome, but just another journal to show you that this is applicable across a number of different journals. This was a journal that I published with ages ago when I was a graduate student, Journal of Cell Science. They're also a not-for-profit journal run largely by volunteers. They do some amazing work. And a student, again, who was defending at the lab next door, had published in the journal, reached out saying, hey, my student is graduating, would you mind doing an interview with the student to highlight the work that they've done as part of their degree? And again, the journal was happy to do that. So again, it's just another sort of marketing opportunity, get your research, get your name out there as much as you can. For graduate students who are looking at including their published work on their CV, I always recommend including things like citation counts, views, downloads, any altmetric score that might be associated with a specific paper, because chances are you may have published the work a year ago, 6 months ago, and it has not had that time to generate a lot of citations. But if you include things like the number of downloads, the number of views, how many times it was mentioned on social media, things like that, again, it's a way to show people outside the academic community that your work has a lot of relevance, has a lot of impact. It's changing the way people are thinking about environmental sciences, conservation, ecology. So this is an example of eDNA that was used to assess sort of bumblebees in the wild. And so you can see again, after it's been published, it's already been cited, downloaded a number of times. You can include these types of metrics when you are adding that type of information to your CV. Creating a digital presence. So graduate students, I think, oftentimes miss this, but a big part of building your portfolio is creating that digital presence, right? You want researchers to be able to find you pretty easily. That's how you kind of build your network, find collaborations, find that next step in your career. So create a Google Scholar account, verify it, make sure that the manuscript that's listed in your Google Scholar is accurate. create a ResearchGate if you can. ResearchGate, I feel like, is becoming more and more popular across academics. It's also a place where you can upload your papers. So other folks who are interested in your work can easily access and read your manuscripts without having to click a bunch of links and find your work elsewhere. ORCID ID is, again, quite popular as well. A lot of funding agencies now require you to have some kind of ORCID ID when you apply for funding. Again, it's just kind of like an online CV. You can list your publications, if you've done any peer review work, awards, affiliations, things like that. And Publons. I think a lot of researchers are already on Publons because it tracks the peer review work that you've done. But as a student, if you've assisted your PI in doing any peer review, make sure you create a Publon so that your work is being tracked, right? Peer review work is valuable to the research community, and it shows that you're engaged with researchers outside of your immediate research area. And then also just getting more out of your research. I find that students spend a lot of time writing the introduction to their thesis, and to be honest, sometimes it's really, really good. I've read a lot of thesis introductions that are quite fantastic, and I was trying to convince them to consider maybe turning that into a short review on a specific topic. So I did so with a couple folks that I knew. One of them, these examples aren't necessarily eDNA related, but a student was studying sort of muscle proteins involved in fruit flies, as well as different proteins regulating cell cycle progression. They were defending, they had already written the bulk of the work in their thesis introduction, and I suggested, hey, if you're open to it, consider reformatting it a little bit, send it to the journal, and we'll see if we can get you another publication out of it. And it worked out great. Another thing is to just really try to avoid leaving parts of your research unpublished. I think as graduate students, you often end up with parts of your project that might not lead to as big a result as you had hoped for, or you were doing preliminary work on a specific methodology, trying to see if it would work for a specific application. It didn't work. But it could still be valuable nonetheless. And so finding a way to publish work that you've worked on is really important. So most journals nowadays have various article types that are dedicated to smaller research sort of findings. So Notes is the one that we use in Genome, but you might have heard of sort of brief reports, short reports, things like that other journals publish. We're typically just looking for a very, very short article, maybe a figure or two, highlighting a very, very small sort of finite research project. So if you have something like that on your desk that you want to get out there, again, happy to consider it at Genome. We don't charge you to publish open access in the journal anyway, so something for you to consider. And then something that I always touch on with both students and more senior faculty members alike is considering the journal as a place to do more than just publishing your research. So journals provide peer review opportunities, guest editing opportunities that students and postdocs can consider. As an example, at one of my journals, we ran a small collection based on a conference in 2025. We had two postdocs who were interested in organizing something like this. So we came together, built a small collection. I think maybe there were 5 or 6 manuscripts that were submitted as part of the conference. They got the opportunity to learn about how peer review was done, how to find peer reviewers, what critiques are valuable when it comes to assessing research. Dr. Shauna has now found her first assistant professor role at Memorial, I believe. And Dr. Marjolijn is still doing her postdoctoral work. But again, there's lots of opportunities for early career researchers, so reach out if you're interested. Journals are also more and more interested in publishing work that has to do with the research community itself, that doesn't necessarily have to be research. So an example that I wanted to highlight is work that was done by a number of graduate students and postdoctoral fellows looking at financial challenges that graduate students face here in Canada. Graduate student funding has always been a challenge for a lot of us, and they came together did an amazing nationwide survey looking at the financial challenges that students were facing. You know, being a graduate student here in Canada, they were looking for a place to share that work and have it be peer reviewed and validated in that kind of a process. So we created space for them in the journal to do that. And you can see it's been cited a number of times, a number of downloads. It was on the news, and you know, the folks who were involved have been interviewed for that work. Again, it's amazing. And then we had them kind of come back and publish another shorter editorial, kind of reflecting on the process about a couple months later. So again, journals are always looking for interesting perspectives when it comes to things that impact the research community. Another great example, I had met a grad student and their PI talking about sort of AI challenges when it came to teaching and research. And they were interested in writing an editorial about it, and I suggested, well, why not one of my journals, right? And so we came together, we wrote an editorial kind of highlighting AI use in academia, you know, the opportunities that AI can provide for us, but also challenges and ethical considerations, especially when it comes to teaching, working with students, training graduate students, things like that. And again, cited a number of times, lots of downloads. So these types of things people care about, right? A lot of us working in academia are looking for insights and information on how AI, funding, graduate student training is impacting the community. A couple more examples. Another amazing researcher, Dr. Aaron Schimmer, met him at another conference last year. We had a great talk about sort of how graduate student training has changed in the last 10, 20 years, right? With new technologies, the expectation of what graduate students are able to produce in a 4 to 5 year span has changed a lot from when I was a graduate student, even just 10 years ago. So again, came together, wrote an amazing editorial kind of reflecting on how having centralized expertise and core services can influence the way a graduate student would learn about techniques and approaches to research. Another researcher that I knew, very passionate about sort of education, rights to education, women's rights, and so we had, again, another article, an editorial really kind of reflecting on access to education in different parts of the world. Lastly, a past collaborator, Dr. Xueqi at Memorial, I believe, was working on hiring practices in her department. So she was one of the only women working in the department. She was brought onto a hiring panel for a number of new sort of assistant professor vacancies. And she was saying, okay, well, how can we approach this in a way that would make the hiring process less biased? Because clearly it's, the composition of the department indicates to us that there may be some biases in the hiring process itself. And so she implemented changes like, having folks' CV and resumes be blinded, so removing names, any sort of racial or gender markers that could bias the hiring committee and the team of folks that were looking to bring in, 5 to 6 new assistant professors. And so as I was talking to her about the work she was doing, I was saying, well, this is amazing. I know that this isn't research, this isn't necessarily work that you were doing as part of your research program, but it's work you're doing nonetheless for the department, and it makes sense that other departments may be interested in hearing about the work that you've been doing here. So why not let us see if we can pull together an article looking at that process and how it's impacting the composition of the biochemistry department. And so again, another great work came out of that. So there's really lots of opportunities to write and share about work that's related to the research community, that's not necessarily research itself, and journals are often quite open to considering work that you may be interested in sharing. So for graduate students, if you're interested in sort of getting involved in writing, outside of just writing a thesis or manuscripts, journals are also always looking for folks who may be interested in working with us for our podcasts and our blogs. So if you're interested, reach out to me, let me know, I'll be here all day. But as an example, we had a student sort of kind of interview Dr. Xueqi that I just talked about to learn more about sort of what got her interested in sort of being a part of the hiring committee, how the lack of diversity in her department had kind of impacted the graduate students on her team, herself as a researcher, and again, we were able to create an amazing kind of blog post and a short kind of interview that you could listen to on our website. So lastly, before I kind of wrap it up, again, Genome, we're completely volunteer run. It's a not-for-profit journal. We love coming to support local conferences. So if you're organizing a conference and you're looking for a little bit of funding, let me know. I'm happy to find a way to make it happen for you. That's what I do. We're also running a collection as part of PISCeS. So if you're interested or you have something on your desk that you want to get off your plate and you want to have it submitted to a journal, reach out, let me know. Again, it's completely free to publish open access in the journal. Happy to make room for your work, whether it's a short research article, a mini review, or even an editorial on how eDNA has impacted your research. Would love to hear it. And that's it.
Thank you very much. No, but some folks in the audience might.
May have thoughts.
We really appreciate your ongoing support of PISCeS.
No, I'm happy to do it.
And particularly giving this workshop today, I think there's a lot of very solid advice, not just for graduate students, but even some of us old graybeards who are somewhat Luddites when it comes to using modern technology. I think we've got plenty of time for any questions that you may have for Calvin, so if anybody wants to pose one. And how do we tag our submissions for the special collection for PISCeS?
There is, so on the website, if you look up Genome, the journal, on the website, you'll find PISCeS as, you'll see the logo, you'll find, there's no way you'll miss it. It'll say PISCeS submissions, you can click into it, and there's instructions on how to submit. So, should be easy. If you have any questions, reach out to somebody, and they'll redirect you to me, and we'll figure it out.
Wonderful, thank you. Any other questions? Yep.
Thanks, Calvin, for the great workshop. That was very interesting. I'm wondering how you see the future of academic publishing unfolding with the rise of generative AI, and how you anticipate that changing the field.
That's a great question. That's tough to answer. I mean, even 5 years ago when I was starting my work in publishing and starting my research in science communications, the use of AI wasn't even a thing, right? Nobody had thought about it. I do believe that AI can be very useful especially for researchers. I've seen a lot of amazing researchers leverage the use of AI for data analysis, for work on interpretation, modeling. So there's definitely use for it. But in terms of manuscript preparation, organizing your references, finding citations, I feel like the bulk of it is still human, at its core, right? But you do need to make sure that when you're citing something that you take a look at what you're citing and make sure, it makes sense and it's accurate, right? There's aspects of research that of course cannot be replaced, right? The role of humans in the way research is done, in formulating research questions, in critiquing the way we do research is a human endeavor, and I don't think AI can really replace it. But of course, I think it's a really tough question to answer. I wish I had an easy, simple sort of solution. But I think as emerging researchers, I would really caution you to avoid using AI in preparing your thesis, in preparing a manuscript, because and ultimately 99% of the time it's not malicious, right? It's a student trying to save time abstract that they think would be stronger and would resonate more with their committee members. But you don't want to be caught in a position where someone thinks that you've used AI, or they know that you've used AI, and they lose trust in the integrity of your work. And you never want to be in that position because it's never true, but it changes the way people see how you do research.
Yeah, so I've been an editor with several journals, and one thing that I always thought about was what efforts are we thinking about doing to translate science to the general public more? Because I know, there's magazines and everything that do that a little bit, but I think like part of the disconnect that we have in the academic community, government industry even, with the general public is that we keep it very close and very technical. And with this, I'm not saying that we have to get away from traditional publications, but I think we have to open spaces where we can translate science. And I know there's blogs and there's very personal efforts that each one of the professors, academics, or PIs do to connect with community, but I think in conferences and in journals, not only because we need to connect better with people, but because public opinion drives policy, and policy drives money into research. So what efforts is, for example, Genome doing in trying to open spaces like this?
That is a great question. I think that's more and more a concern for a lot of us researchers. We're very reliant on public support for funding, right? All of my funding sources or national funding sources. I think journals do need to do more and more, and I think regardless of where you choose to publish, I always encourage you to kind of push the journal that you're choosing to publish with to see what they can do, what avenues they have to kind of get research out there, get research seen by folks that aren't always researchers, right? So yes, so Genome runs a blog. We also submit press releases. So if you submit to Genome and we flag that your work may be of interest to news sites, we will prepare a press release and get that out there for you. We're also quite engaged with various sort of science communications programs, but even those are, I think, sometimes geared towards early career researchers, high school students, not necessarily the lay public. So to answer you, I feel like the journal could do more, but it is a volunteer-run team, so there are limits. If you have ideas, let me know. If you're involved in something that you think Genome can support in terms of public outreach and public engagement, let me know. We'd love to support it. There's actually an article published in Genome 2-ish months ago that was an editorial kind of reflecting on exactly what you've been talking about. But the bridge is hard, right? A lot of times, even when science is on the news, even when science is shared in a way that we think the public would be interested in reading about it and learning about it, it doesn't always resonate the way we want it to, right? Or they're picking apart pieces that they think clicks when it's not the picture that we want to present. Again, I don't know. I don't have an easy answer. It's tough, but I think everyone on the team is trying their best. If you do have ideas, suggestions, let me know, because I do want to hear them. I really, really do. All right, I'll pass this back to you.
Thank you, terrific. Let's give a big round of applause for Charlyn. All right, we've been very deliberate at having generous times for break, so please follow the instructions you were given last night at the reception. For those who were there and for those who weren't, I'm going to reiterate them. Find someone you don't know and talk to them. And we'll see you back at 11 for our next keynote presentation.